Hello everyone, welcome to the new session of genetics and plant breeding. So in this session we are going to study about the cell and its structure. This is the first part of the cell and its structure. Okay, so what is cell? So let us find out what is cell. Anything less than a complete structure of a cell does not ensure the independent living. So, if a particular entity which is less than a cell structure, that will not be a living entity, right? So, cell is the fundamental structural and functional unit of all living organisms. So, this is the definition. This is the definition of the cell. So, cell is the fundamental structural as well as the functional unit of all living organism, right? Okay, so what Anton von Leeuwenhoek first saw and described the living cell. So he was the scientist who first saw and described about the living cell. Robert Brown later discovered the nucleus. He discovered nucleus in the cell, that in the center there is a nucleus. There is a nucleus. So this was discovered by Robert Brown. The invention of the microscope. So when there is a invention of the microscope, and also after the invention of electron microscope, right? Electron microscope. It revealed all the structural details of the cell. So this microscope becomes the aid to see the internal structure of the cell. So when there is a discovery of the microscope, okay, and also the higher grade of microscope, that is called as the electron microscope, the inside structure of all the cell can be seen, right? All the organelles, and inside the organelles also the details of each and every organelles can be seen with the help of electron microscope. Okay, so after the discovery, after the invention of such microscope, so it was very helpful to study the structural detail of the cell. Okay, now we will study about the cell theory. This is a very important topic. In 1838, Matthias Schleiden, who was a German botanist, he was a botanist, okay, and observed that all plants are composed of different kinds of cell which forms the tissue of the plant, right? So we can see the different types of plant cell in one individual plants only, right? Theodore Schwann, on the other hand, he was a British zoologist. He reported that the cells had a thin outer layer known as the plasma membrane, right? So Schwann was studying about the animal cell and Schleiden was studying about the plant cell, right? So on his studies on plants tissue, he concluded that the presence of cell wall is a unique character of the cell. Okay, so Schleiden was studying about the plant cell. So he concluded that the presence of cell wall is a unique character of the plant cell because this cell wall is present only and only in the plant cell, right? So on the basis of this, Schwann proposed that hypothesis that the bodies of animals and plants are composed of cells and products of the cell, right? So both came with a conclusion. Now, Rudolf Virchow in 1855 first explained that the cell divided and new cells are formed from the pre-existing cell. So Virchow said that if there is a cell, okay, and new cells are formed. So this new cell will form from the pre-existing cell only. So there is a cell division. When the cell divide, first the nucleus divide into two, right? Like this. And later cytoplasm also divide and form two individual cells, right? As we see in the case of amoeba, binary fission, at the time of binary fission, even in the case of bacterial cell, right? So omnis cellula e cellula, it means that new cells are formed from the pre-existing cells. This was the statement given by the Rudolf Virchow. So in the examination, it can be asked that who stated that new cells are formed from the pre-existing cells. Okay, so the name can be in the option can be given Rudolf Virchow, Schleiden, C. Schwann, and D. Robert Brown. So who gave that? Rudolf Virchow. So this particular point is very, very important. He modified the hypothesis of Schleiden and Schwann to give a cell theory in a final shape. So earlier, Schleiden and Schwann gave this cell theory, but later Rudolf Virchow modified this hypothesis, and what it came in a form. So cell theory as understood today is all living organisms are composed of cells, right? So whether it is a plant cell or whether it is an animal cell, it is composed of cell and also the products of cell, right? All cells arise from the pre-existing cell. This is very, very important. So new cells are formed from the pre-existing cells only. So this was the modified form of the cell theory given by the Adolf Virchow. The onion cell, which is a typical plant cell, right? So we generally, when want to see a plant cell, we take onion peel from the scale leaves of the bulbs, right? So it is a typical plant cell and has a distinct cell wall as its outer boundary and just within its cell membrane. So if we want to see an onion peel cell, then we can see the plant cell like this. This is the cell wall. This is the cell wall, and this is the cell membrane. Cell membrane, right? The cell of the human cheek have an outer membrane as the delimiting structure of the cell. So human cheek cell, we are talking about the human cheek cell. So human cheek cell is surrounded by a plasma membrane only, or the cell membrane only. Cell membrane only. But in the case of onion peel cell, we can see that the outermost delimiting structure is the cell wall, and then comes the cell membrane, right? Yes. Inside each cell is a dense membrane bound structure called nucleus. So in every cell, whether it is a plant cell or an animal cell, we can see inside the cell that in the center there is a dense membrane bound structure which is called as nucleus. So this nucleus contains the chromosome, which in turn contain the genetic material. That contains the genetic material, right? So inside this nucleus, genetic material in the form of chromosome is present. Okay. Cells that have a membrane bound nuclei are called eukaryotic, whereas cells that have lack membrane bound nucleus are called prokaryote. So if we draw a eukaryotic cell, then it will have a membrane bound nucleus. So this is the nucleus. This is the nucleus, which is a proper membrane bound. But if we see the structure of a bacterial cell, then there is no membrane bound structure. The chromosome is present in the cytoplasm itself. Okay, and this is called as nucleoid. This is called as nucleoid. This is not a true nucleus. So this is in the case of prokaryotic cell. In both prokaryotic and eukaryotic cell, a semi-fluid matrix called cytoplasm occupies the volume of the cell. So this cell is composed of a semi-fluid matrix, whether it is a prokaryotic cell or a eukaryotic cell. Though this semi-fluid matrix is called as cytoplasm. Right? This is the eukaryotic cell and this is the prokaryotic cell, right? So this cytoplasm is what? This is the semi-fluid matrix, and all the organelles in the eukaryotes are present in this matrix only. Okay. The cytoplasm is the main arena of the cellular activities in both plants and animal cells. So we are talking about the plant and animal cell, then it means that we are talking about the eukaryotic cell. So all the activities are taking place in the cell in this cellular matrix only, right? Various chemical reactions occur in it to keep the cell in the living state. So all the living activities are occurring inside the cytoplasm. Beside the nucleus, the eukaryotic cell have the other membrane-bound organelles like the endoplasmic reticulum, Golgi complex, lysosome, mitochondria, microbodies and vacuoles. These are all membrane-bound organelles and they are present in the eukaryotic cell. These organelles are not present in the case of prokaryotic cell. So the prokaryotic cell lacks such membrane bound organelles, right? So this is one of the major difference between the prokaryotic cell and the eukaryotic cell, right? So beside nucleus, eukaryotic cells are having different membrane bound organelles like endoplasmic reticulum, Golgi complex, lysosome, mitochondria and microbodies, right? But these organelles are not at all found in the case of prokaryotic cell. Ribosome and non-membrane bound organelles. Ribosomes are this is one of the organelle which is not having any membrane structure, right? So membrane is absent in the case of ribosome. So sometimes it can be asked in the examination, which of the following is lack any membrane, or which of the following organelles do not have any membrane. So the option can be lysosome, mitochondria, chloroplast and ribosomes. So the ribosome will be the correct answer, right? Okay. So ribosomes are non-membrane bound organelles found in all cells, both eukaryotic cell as well as the prokaryotic cell, right? So it is present in both eukaryotic as well as the prokaryotic cell, but there is a major difference between the ribosome of eukaryotic cell and the prokaryotic cell. What is that? So ribosome in the case of prokaryotic cell is of 70S type and ribosome of eukaryotic cell is of ATS type. This is the difference between the ribosome of eukaryotic and prokaryotic. Okay. So within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles like chloroplast in plants and mitochondria and on rough endoplasmic reticulum. So inside the two organelles in the case of eukaryotic cells, inside the two organelles, that is the chloroplast and mitochondria, ribosomes are present. Whereas in the case of endoplasmic reticulum, rough endoplasmic reticulum, ribosomes are just attached to its surface. They are just attached to its surface. But they are present inside chloroplast and mitochondria. So we will study these all things in a detail when we will study about the cell organelles. Animal cell contain another non-membrane bound organelle called centrosome, which helps in the cell division. So this centrosome is not found in the case of plant cell. It is only found in the case of centrosome and it helps in cell division. Cells differ greatly in size, shape and activities. There are different size and shape of these cells, because they have to perform different function, and according to that their size and shapes are adapted. Okay, so for example, mycoplasma, the smallest cells, and they are only 0.3 micrometer in size. The largest isolated single cell is the egg of an ostrich. This is the largest cell and mycoplasma is the smallest cell. Among the multicellular organism, human red blood cells are about 7 micrometer in diameter. So this is the red blood cell. Its diameter is around 7.0 micrometer. Nerve cells are some of the longest cells. So this nerve cell is the longest cell. So here we can see the nerve cell. This is the longest cell in the human body. Cell may be disc-like. Now we are talking about the different shape. Cell may be disc-like, polygonal, columnar, cuboid, thread-like and even irregular. So why they are having different shapes? Because they have to perform the different functions. The shape of the cell may be vary with the function they perform. So we discussed about this just now, that cell has to perform different functions. So according to that, they vary their shapes. Prokaryotic cells represented by bacteria, blue-green algae, mycoplasma or PPLO. So this is one of the example of mycoplasma, PPLO, pleuropneumonia-like organism, full form of it, PPLO. Okay, so these are the different examples of prokaryotic cell. So when we ask the examples of prokaryotic cell, then bacteria. This is one example. Blue-green algae or cyanobacteria, another example. Mycoplasma, another example. So these are the different prokaryotic cells. Okay, they are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. So these prokaryotic cells are also having different shapes and size. So basic shapes of bacteria are bacillus. So bacillus bacteria are rod shape. This is the rod shape bacteria, that is bacillus. Coccus, spherical shape. So Diplococcus, two coccus, okay, like this. Vibrio, comma-shaped bacteria, like this. Spirillum, spiral-shaped bacteria, spiral shaped bacteria. So bacteria also having different shapes. The organization of the prokaryotic cell is a fundamentally similar, even though the prokaryotic exhibit a wide variety of shapes and functions. So prokaryotic cells are fundamentally, because this is also a cell, right? But they are fundamentally similar to the every cell, but they are having variety of shapes and functions. All prokaryotic cells have a cell wall surrounded by the cell membrane except mycoplasma. So mycoplasma do not have any cell wall, but other prokaryotic cells are having cell wall as well as the plasma membrane. The fluid matrix filling the cell is the cytoplasm. So cytoplasm is present in the case of both eukaryotes and prokaryotes. There is no well-defined nucleus. We have already discussed that if we talk about the nucleus in the prokaryotic cell, then the chromosome is present naked. Means it is not bound with any membrane-like structure. So nucleoid region, it is called as nucleoid or simply nucleoid region. Okay. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have a small circular DNA outside the genomic DNA. So beside this circular DNA, this chromosome, beside this one chromosome, which is the main chromosome of the cell, there are some circular DNA. These circular DNA are called as plasmid. Okay. So these smaller DNA are called as plasmid. The plasmid DNA confers certain unique phenotypic characters of such bacteria. So these plasmid contain simply one or two genes. Okay. They contain less number of genes as compared to the main chromosomal DNA. One such character is resistance to the antibiotics. So these plasmid contains genes for the antibiotics. Okay. So this is very, very important. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. So when we will study about the biotechnology, then this plasmid is used as a vector. We will study there that this plasmid is used as a vector. When we have to transform any cell by transposing any foreign DNA inside the plant cell or animal cell, then these plasmids are used as a vector. So there we will study about those structure. No organelles like the one in eukaryotes are found in the prokaryotic cell except for the ribosome. So ribosomes is the common organelle which is present in the case of prokaryotic cell and eukaryotic cell, but still there is a difference in the type of ribosome. Prokaryotes have something unique in the form of inclusions. They are essentially enfolding of the cell membrane. So there are some inclusion bodies, and also at some region the cell membrane get enfolded, and that forms the mesosome structure. Okay, now we will talk about the eukaryotic cell. The eukaryotes include all the protists, plants, animals, and fungi. So these are all the categories of the eukaryotic cell: protists, plants, animals, as well as the fungi. There is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. So here we can see a definite membrane-bound organelles are present in the cytoplasm of the eukaryotic cell. So they form the compartment, small compartment, and that is very unique for its function, right? Eukaryotic cell possesses an organized nucleus with a nuclear envelope. So they are having organized nucleus. Organized nucleus means they are having a proper covering. They are having a proper covering that is called as nuclear membrane or nuclear envelope. So this is totally absent in the case of prokaryote. Okay. So in addition, eukaryotic cell have a variety of complex locomotory and cytoskeletal structures. So those structures are absent in the case of prokaryotic cell. Their genetic material is organized into chromosomes. So in the case of eukaryotic cell, the genetic material in the case of eukaryotic cell, they are present in the chromosomes, different chromosomes, right? But in the case of prokaryote, a single circular DNA is present. Single circular DNA is present. Single circular DNA is present in the case of prokaryotic cell. Prokaryotic cell. Okay. Here we are having number of chromosomes. Number of chromosomes in the case of eukaryotic cell. This is the difference. This is the difference. And this is the nuclear membrane. So that nuclear membrane is not found in the case of prokaryotic cell. All eukaryotic cells are not identical. There are so many differences in the eukaryotic cell. The major difference, we can see that plant and animal cells. They are different as the former possesses cell wall. So in the case of plant cell, we are having cell wall, plastids, and a large central vacuole, which are absent in the case of animal cell. So if we talk about the plant cell, they will have a very important wall that is called as cell wall made up of cellulose. So this is totally absent in the case of animal cell. Plastids are also present and a central vacuole, large central vacuole is present. On the other hand, animal cell have centrioles which are absent in almost all plant cell. Okay, vacuoles are also present in some of the animal cell, but their size are very small, right? So this is the difference between the plant and animal cell. Here we can see the plant and animal cell. So they are having cell wall, okay, and inside the cell wall they are having plasma membrane as well. And in the case of animal cell, we will have only outer covering that will be of plasma membrane, right? So here we can see the central vacuole, this central vacuole. Okay. So there are so many differences. Centriole is present in the case of animal cell, but this structure is absent. Okay. Rest of the structures, they are also having the nucleus, This mitochondria is present in the animal cell. Okay. Mitochondria is also present in the plant cell. Chloroplast. Chloroplast is present in the plant cell, but this is totally absent in the case of animal cell. Okay. Endoplasmic reticulum, both the types are present in the both the plants as well as the animal cell. Okay. So this is all about the first session on the cell and its structure. We will have some more session to complete and study about the different cell organelles of the cell. Okay. Thank you very much.